Hi and welcome to our tutorial. This is a 3-video series on immunology and cancer. So this is the first part. Before we proceed, kindly like, share and also subscribe to the channel. Our immune system protects our body against invading pathogens such as bacteria and virus, and also internal factors such as cancer. A key path is normally evoked by special cells of our immune system known as dendritic cells. These cells penetrate intracellular spaces to kill antigens. An antigen may originate from bacteria, viruses, or cancer cells. Proteins from cancer cells contain many mutations. So when a dendritic cell ingests proteins from cancer cells, it manufactures short sequences of amino acids known as antigens or neoantigens if they originate from cancer cells. The cell then loads this antigen into histocompatibility complex. This refers to protein molecules that are found on the cell surface. We are going to look at histocompatibility complexes in a few seconds to come. The dendritic cell then travels into the lymphatic system. Once there, it will then present the neoantigens or antigen that it has found on other key components of the immune system. Let's look at major histocompatibility complexes. So what are they? These are important genes that encode proteins on the cell surface. They have an important role in the immune system. So they come in two forms. The first form is MHC1, that is major histocompatibility complex 1. And the second form is MHC2, that is major histocompatibility complex 2. So what are the importances of major histocompatibility complexes? The first importance is antigen presentation. MHCs display peptides on the cell surface for recognition by T cells. So this is an important process of an immune system response for destroying invading pathogens. Importance number 2 is cell self-recognition. Presence of MHCs on the cell surface prevents the immune system from targeting its own cells. Certain MHC alleles are associated with increased risk of autoimmune diseases such as Hodgkin's lymphoma and multiple sclerosis. The third importance of MHCs is tissue allorecognition. This is an important factor in preventing a successful organ transplant. Allorecognition is the ability of an organism to distinguish its own tissues from the tissues of another organism from the same species. This can lead to rejection of a transplanted organ. What are the mechanisms of allorecognition? You need to know that when an MHC molecule is loaded with an antigen, or what we call a peptide or a protein, it forms MHC protein complex. So this MHC protein complex normally has a determinant that T cell normally recognize. So this determinant is known as an epitope. So an epitope can either be self or non-self. Now, antigens from transplanted cells are displayed as non-self. Now, to prevent an allo response from non-tolerant recipients, immunosuppressive drugs are normally administered, but they are known to have a long-term effects. So there are two major properties of MHCs that make it very difficult for a given pathogen to evade immune response. So the first property is they are polygenic. 1 and MHC2 contain various different classes of genes. So this one enables every individual who has got MHCs, that is MHC1 and MHC2, to have a variant types of genes within the same species with peptide binding specificities. Another property is they are highly polymorphic. There are multiple variants of genes within the population as a whole. MHC genes are the most polymorphic genes ever known. So what are the major differences between MHC1 and MHC2? MHC1 presents antigens that are intracellular or endogenous. MHC2, on the other hand, presents antigens that are extracellular or exogenous. Another difference is that MHC1 contains alpha chain and beta 2 microglobulin. MHC2, on the other hand, contains alpha chain and beta chain. In MHC1, cell surface expression is ubiquitous. That is expressed in most nucleated cells. In MHC2, cell surface expression is restricted to antigen presenting cells, that is dendritic cells, macrophages and beta cells. Under MHC1, the source of antigen is intracellular pathogens. Under MHC2, the source of antigens is extracellular pathogens. Under MHC1, antigen processing is through endogenous pathway. That is, antigens derived from within the cells. Another difference between MHC1 and MHC2 is the size of peptides. MHC1 generally presents smaller peptides, that is 8 to 10 amino acids. MHC2, on the other hand, generally presents larger peptides, that is 13 to 25 amino acids. Another difference is the T-cell interaction. MHC1 interacts with CD8 T cells, also referred to as cytotoxic cells. MHC2 interacts with CD4 T cells. These ones are also known as helper T cells. Now let's look at T cells. T cells undergo maturity in the thymus gland, which is the central half of the lymphatic system. Through a complex selection process, T cells develop highly specific receptors known as T cell receptors or TCRs. Our bodies can generate millions of TCRs, giving us enormous potential to fight a vast variety of pathogens and cancer cells. Within the thymus glands, T cells are normally tested against our body's database as a representative measure against autoimmunity. Now, if a match occurs, the T cells and the self-reactive TCRs are assured together. T cells surviving to maturity are differentiated into CD4 and CD8 co-receptors. Now, at this point, T cells are ready to exit the thymus gland and begin circulating within the blood circulatory system and the lymphatic system. Moving around increases the chances, that is the probability that a T cell will meet its target antigen. The dendritic cell then presents the antigen on the Mhcs and therefore enabling T cell recognition. After some time, a dendritic cell finds a T cell bearing TCR compatible with the antigen it is representing. The T cell then binds to the antigen MHC complex, thus initiating the process of T cell activation. We can go through our notes. Our immune systems defends our bodies against external factors such as invading pathogens and internal factors such as cancer cells. A key path is especially caused by dendritic cells. These are special immune cells. These cells penetrate extracellular space to kill antigens. An antigen may originate from foreign materials such as virus, bacteria, or cancer cells. So on the diagram, I have illustrated a dendritic cell. This is a dendritic cell trying to ingest a pathogen. Proteins from cancer cells contain many mutations. The dendritic cells then process mutatic proteins, producing short sequences of amino acids called antigens or neoantigens if they originate from cancer cells. The dendritic cell then loads this antigen or neoantigen into major histocompatibility complex, that is MHCs, found on the cell surface. So histocompatibility complexes come in two forms, that is MHC1 and MHC2. The antigen-loaded dendritic cell then travels to the lymphatic system. Once there, it will present the neoantigens or antigens it has found on other key components of the immune system. Major histocompatibility complexes. So this is a group of genes that encode proteins on the cell surface and have an important role in the immune system. They come in two forms, that is MHC1 and MHC2. Importances of major histocompatibility complexes. Number 1, antigen presentation. They display peptides on the cell surface for recognition by T cells. So this is an important process of immune response for destroying invading pathogens. Importance number 2 is cell self-recognition. The presence of MHCs on cell surface prevents the immune system from targeting its own cells. Certain MHC alleles are associated. Importance number 2 is cell self-recognition. Presence of MHCs on cell surface prevents the immune system from targeting its own cells. Certain MHC alleles are associated with an increased risk of autoimmune diseases such as Hodgkin's lymphoma and multiple sclerosis. Then importance number 3 is tissue allorecognition. An important factor in prevention of successful organ transplant. Allorecognition is the ability of an organism to distinguish its own tissues from those of another organism within the same species. It can lead to rejection of a transplanted tissue. Mechanism of allorecognition. T cells recognize determinants on the donor MHC molecule peptide complex displayed at the cell surface. This is because MHC molecules display an antigenic determinant called an epitope. An epitope can either be self or non-self. Antigens from transplanted cells are recognized as non-self. To prevent an allo response in an intolerant recipient, immunosuppressive drugs are administered but are known to cause long-term adverse effects. So what are the two important properties of MHCs that make it difficult for pathogens to evade immune response? So the first one is polygenic. They contain several different MHC class 1 and MHC class 2 genes, so that every individual possesses a set of MHC molecules with different ranges of genes with peptide binding specificities. They are highly polymorphic. There are multiple variants of each genes within the population as a whole. MHC genes are the most polymorphic genes. So what are the major differences between MHC1 and MHC2? Major histocompatibility complex 1 has the following features. Presents antigens that are intracellular or endogenous. Presents antigens that are intracellular or endogenous. Under MHC2 presents antigens that are extracellular or exogenous. Point number 2, it contains the alpha chain plus beta microglobulin. This one contains alpha chain and beta chains. MHC1 cell surface expression is ubiquitous, that is expressed on most nucleated cells. Cell surface expression is restricted to antigen-presenting cells, that is dendritic cells, macrophages, and beta cells. Then under MHC1, the source of antigen is intracellular pathogens, that is viruses or intracellular bacteria. Under MHC2, antigen source is extracellular pathogens, that is bacteria and parasites. Then go to the next point. Antigen processing is through endogenous pathway. That is, antigen derived from within the cell are processed and presented on MHC1 molecules. Under MHC2, antigen processing is through exogenous pathway. So antigens which are internalized from extracellular environment are reprocessed and presented on MHC class 2 molecules. Peptide size. Major histocompatibility complex 1 presents small peptides, that is 8 to 10 amino acid residues. MHC2 presents large peptides, that is 13 to 25 amino acid residues. We can also differentiate them in terms of T-cell interaction. They interact with the CD8 T molecules, that is under T-cell interaction, MHC1 interact with CD8 T cells, that is cytotoxic cells. And MHC2 interact with CD4 T cells, that is helper T cells. The co-receptor is the CD8 molecule, that is under MHC1. Then the co-receptor is CD4 molecule under MHC2. Immune function is cytotoxicity under MHC1, while under MHC2 we can say that the immune function is coordination and activation of other immune cells. Then under location, we can say that under location, we can say that MHC1 is present on all nucleated cells, while MHC2 is present primarily on antigen-presenting cells. So they undergo maturation in the thymus gland, that is the central half of the lymphatic system. Through complex selection process, T cells generate a highly specific T cell receptors, that is TCRs. Our bodies can generate quadrillions of distinct TCRs, giving us enormous potential to fight a great variety of pathogens and cancer cells. Within the thymus gland, TCRs are tested against our body's database or self-sequence as a representative measure against autoimmunity. If a match occurs, the self-reactive TCR is assured together with T cell itself. T cells surviving to maturity are differentiated into two types, CD4 and CD8 co-receptors, often referred to as a helper and killer T cells respectively. At this point, T cells are ready to exit the thymus gland and begin circulating within the lymphatic system and the bloodstream. Moving around increases the chances that a given T cell will meet its target antigen. Dendritic cell then presents the antigens on the Mhcs, thus enabling T cell recognition activity. After some time, a dendritic cell finds a T cell bearing a TCR compatible with the antigen it is presenting. T cell then binds to the antigen MHC complex, initiating the process of T cell activation.